Um, Welcome to the March Statistics Seminar on the last day of winter. We're going to talk about observational studies today, and in particular look at doing comparative effectiveness studies. That is where we're going to compare treatment groups, medications, or whatever you might want to study. And in particular, we're going to consider propensity scores and how we use these in observational studies. Let's see if I can get here. Okay, so I'll talk a little bit about study designs, and then of course in observational studies the main problem is selection bias, and then we're going to look at propensity scores, how they're calculated, what they mean, and then I'm going to use two studies to illustrate. The first one being propensity score matching, and then the second one being what's known as inverse probability weighting, where we use propensity scores as the inverse of the propensity score. So in a randomized clinical trial is really the gold standard of study designs, because randomization potentially or supposedly equalizes potential confounders in the treatment groups. So whatever confounder there might be, it's sort of washed out by the randomization procedure. This is, as I mentioned, the gold standard for research designs, but they do have some limitations. One, of course, being that they're quite a bit more expensive than observational studies. And in particular, if you follow drug studies, you know that they're very, very expensive. And then also there's the argument that you have limited data. That is, it's on a specific sample from a population, and you have people that obviously volunteer to be in the study, even though they might be at some point randomized to a particular treatment group. Observational studies, on the other hand, are a little more easier to do, and that is that you may your data may already exist, like in an electronic medical record. Also, electronic medical record might contain more information than you'd be able to collect in a randomized trial. However, the primary drawback, of course, to observational studies is selection bias. And what we mean by that is that people are self-selected, or they're selected by someone else in a particular for a particular treatment group. They're not randomized, and they're not either because there's an ethical issue or because it's just impractical, or it would be just impossible to randomize in a particular situation. So there are different ways that have been used to, or even are used to, reduce selection bias. And one of those is just matching by different covariates that you have in your data set, age, race, sex, so on. However, only a few of these are to include are realistic. And so, for example, if you have categorical variables, just for example, like race and sex, depending on how many categories you have, the number of categories, for example, you know, female, black, female, white, female, Hispanic, and then you have, male with the race categories. Pretty soon you get so many categories that you're not going to have enough patients or enough subjects in your study to fill all of those, or you end up with only very, very few. And then if you have continuous variables, say like age, then the question is, well, how close are you going to match? So for example, on age, you're going to match within 2 years, 1 year, 10 years. And of course here, the closer the match or the tighter the match, then the fewer matches you're probably going to end up having. Whereas if you have a wide age range to match, like 10 years, you'll obviously be able to match. But then, of course, the question comes, well, is that really a match? Is a difference of, say, for example, 10 years in age, are they really comparable? And then, of course, another way is just to put the covariates that might be confounders in a regression equation. And then the regression equation would have as one of its variables, one of its covariates, the treatment group. And again, this has some limitations because depending on your sample size, you would have a limited number of variables that you could include in the regression model. So the current thinking about reducing selection bias is to use propensity scores. And there's some other ways of doing this, but the two primary ones that are used are propensity score matching and then inverse probability weighting. And so a propensity score is the concept or the idea that you have a propensity to be in one treatment group or another, where the group assignment could be assigned or even randomized. I want to make a point here is that you can't be assigned to a comorbidity or risk group. For example, you can't compute propensity scores on whether or not a patient has diabetes. That's a risk factor that person has. You can't be assigned that. Interestingly, though, I've seen studies where they do this. They create a propensity score to be in diabetes or body mass index, BMI, group, but that's inappropriate. And so the way that you calculate the propensity score is with logistic regression, which we've talked about earlier, with the treatment group as the outcome. And so I'll just use A or B here as an example. And so you have as your outcome A or B, yes or no, 1 or 0. And then the predictor variables are all the demographic, clinical, laboratory variables that you have in your data set. And you use those to predict which group a person would be in. The one variable, though, that you can't use in your logistic regression equation is the outcome of interest. So if you're interested in survival rates, for example, you can't use that as the in your logistic regression. And so the thinking is that in generating a propensity score that we're going and assigning people based on their propensity score to one group or the other, that we come out with a quasi-randomized study. That is, that we're going to use it to an attempt to balance out potential confounders between patients. So the propensity score, of course, is you solve the logistic equation for each patient. And so then you can calculate a probability. So if we want to calculate the probability for group A, and our probability being the exponent of the logit equation divided by 1 plus the exponent of that equation, then so the probability of being in group A is, and then if the probability of being in group B is just 1 minus that. So we can, depending on which group you want to focus on to be as your primary treatment group, or your treatment group versus a control or whatever, you solve the logistic equation for that particular group. And so the other group then is the probability of being in that group is just 1 minus the probability of the other group. So in propensity score matching, what we want to do is match a patient in group A and a patient in group B with the same propensity score. Of course, not everybody's going to have exactly the same propensity score. So the question is, how close does the propensity score have to be for us to call it a match? And so typically what is done is we set a criteria for a match. And then a term that's used is the term greedy matching. That is, we're going to set a criteria for match, and then everyone who has a smaller criteria will be included for a match. So here's an example. And so I've calculated a propensity score for patients in A and B with 8 decimal places. And so the first, in the first patient, they're A and B, you see they have exactly the same propensity score. So we've matched on all 8 digits. In the second one, you can see then that we've matched on 7 of the 8 digits. And in the third one, we've matched on 6 of the digits, and then and so on, until we get down to the last last patient here, 0.6, where we've matched on just 3 digits. So the question is how close of a match is acceptable. And so, as I mentioned, this is assumed to most closely mimic a randomized trial. However, as you might expect, not everyone is going to be able to be matched. In other words, we won't be able to match a certain number of patients. And it turns out that this is in almost every study that does propensity score matching, that you will find that about a third, 30% or greater of the patients can't be matched. Okay, so the second one that I'm going to talk about is inverse probability weighting. And so again, we have the propensity score, the probability of being in one group or the other. And so if we have our 2 probabilities, our probability of being in group A and then the probability of being in group B is 1 minus the probability of A. And we have our sample size of n, certain number of patients. And then we have the treatment indicator of whether they're in group A or group B. And then we have some response variable we'll just call R. Then the inverse weighting estimator is just for each group, the response minus the mean of the response in that group divided by the propensity score of that group, being either the probability of being in group A or the probability of being in group B. Now, most statistical software just requires the propensity score, and they'll do the calculations for you in a sense. Calculating the propensity score is really straightforward, really easy. You don't really need to, for the most part, worry about doing anything other than that. So the advantage, of course, of inverse weighted propensity scores is that it uses all patients, because all patients will have an IPW, an inverse propensity score. You can use it for censored response data. The problem, though, with propensity scores, and this is true of matching as well, is if you have an unmeasured confounder, that is, you've calculated your propensity score with all of the demographic, clinical, lab values that you have, but maybe there's a variable that you didn't measure, that you didn't have in your data set, and it turns out that this variable is the confounding variable. And so in that case, then your results of your propensity score analysis may not be correct, and we'll talk about that later on. Okay, so the first study I want to present is a study that was an intervention to improve neonatal outcomes. And so basically, there were outcomes before the intervention, outcomes after the intervention. It's obviously not a randomized trial. And then, of course, obviously, again, you're not likely to have the same women before and after the intervention participating. So here's kind of a picture, a schematic of what we have. So the data for before the intervention was from 2004. The intervention occurred during the year of 2005, and then the women giving birth after the intervention in 2006. So basically the comparison is for women before the intervention versus women after the intervention. And so in terms of the data set that we had, it was quite large. So there are some 5,500 women before the intervention and 4,700 giving birth after the intervention. So that, you think, wow, you got 10,000 individuals in this study. However, of course, not every woman in the 2006 cohort participated in the intervention in 2005. So it turns out that only about 40% actually participated in the intervention. So here are the variables that we use to calculate the propensity score. So our logistic regression was before the intervention, 2004, and after the intervention. And so you can see that there's quite a large number of variables that we're going to use in our logistic regression equation. And so, I say, well, that's a lot of variables to put in a regression equation. Well, it sort of is, but remember what we're doing here is we're not calculating we're not computing a prediction equation that we want to use on a different sample. We're wanting to match as closely as possible the individuals, the participants within our sample. So in this case, we'll put in as many variables in the regression equation as we can. And so you see that there are a lot of here, of course, related to birth, to mother variables, delivery variables, and so on. So it turns out then that if we took a matching criteria of 0.0001 or less to be called a match. And so out of the 10,000, we had almost 7,000 patients, or 3,500 pairs, but because not all participated, then we had to discard quite a large number, and it turns out that we ended up with only 1,400 intervention pairs. okay, or about 41% of all of our total available patients in that time period. So here's an example of just a few of the data points in our data set. And so what I have here is the intervention patients in 2006, and then the prior intervention patients in year 2004. And I've calculated the difference in their propensity scores. And so you can see that there's quite a few here that are, or all of these here are less than our cutoff criteria of 0.0001. So the problem, though, is that as I mentioned before, not all of the women giving birth in 2006 participated in the intervention. So if we look at this, then here's a patient where we had a really good match, but the intervention person actually didn't participate in the intervention, and so on. You can see here that there's quite a few out of just this small subset here that actually didn't participate in the intervention. And so that's why we come up with only 1,400 and some pairs. So since this is basically then a pre-post matched pairs design, we have, of course, the intervention, no intervention. That's what we call a between subjects factor. But the pre-post difference is what we call a within subjects factor, because that's correlated data. So hopefully our simulated randomized trial will balance out the potential confounding factors, all of the factors that were in our table that we looked at that was included in our regression equation. Okay, so obviously this is not perfect, you know, by any means, but it's, you know, it is what it is. So it's our attempt to balance out to make the cohorts, the 2004, the pre-intervention and the post-intervention cohorts, more or less equal with respect to potential confounding factors. So since this is correlated data, we could just do a paired t-test, or if we set our data up a different way, we can use a general linear model and basically amount to a repeated measures analysis of variance. So here is the paired t-test results. So it's just a difference in the year 6 birth weight, which was just one of the outcomes we were interested in. I just use this as an example, comparing that with the year 4 birth weights. And so you can see here that the year 6 birth weight is 61 grams higher than the year 4 birth weight, which even with our reduced sample size, we still have, you know, 1,400, you know, observations. So in each intervention period. So the difference is, you know, statistically significant without much of a problem. If we want to do this in a general linear model way, then we can restructure our data this way, where we have the intervention and the pre-intervention just designated by the year here. And then we have a ID that links the two patients together. Even though these are different patients, they're linked by their propensity score. And so then we can just do a general linear model sort of analysis. And so you can see that the results we get here are the same as in our paired t-test, where 61 was the difference between the 6-year and the 4-year, and then our constant here, if it's the 6-year. If it's just the 4-year, then the mean birth weight was 3,434 grams. So that's a way of doing the analysis of a non-randomized study where there's a lot of potential for confounding. And there's the results that we got in our Ttest. Okay, so the second one that I want to focus on is using the IPW, inverse probability weighting. And so in this study, what we did was to compare effectiveness of revascularization strategy in specifically comparing percutaneous coronary intervention versus coronary artery bypass. And so this was a fairly complicated study, not just the analysis, but just getting the data together. So it linked two different data registries, the ACC National Cardiovascular Data Registry, that's the American College of Cardiology, which would be the PCI patients, and then the Society for Thoracic Surgeons Adult Cardiac Surgery Database, which would be the CABG patients. And so we got data from the years 2004 through 2008. And so obviously there's going to be differences here that we're not going to be able to control for by use of randomization. So we're going to use propensity scores and inverse probability weighting. So this is quite a large database, 1.5 million PCI claims and over 500,000 CABG claims. And not only that, it's, these are data, since their registries are coming from different sites. After the exclusion criteria, then we ended up with a total of 103,000 patients, 86,000 were PCI patients, and the other, 86,000 CABG patients, that's 15% of the total, were included in the analysis. And I have this paper as a reference if you're interested in looking at it. So that's the database that we came up with, a fairly large database. So obviously there's going to be differences between the PCI and the CABG populations. And so a large amount of baseline variables were collected. But of course, there's always the problem here when you're wanting to compare outcomes from different databases. You may not always have the same variables available. So obviously we had to use variables that were common to both registries. And so the propensity scores were calculated on the basis of patient and hospital characteristics. And then we did our logistic regression with CABG as the intervention of interest, and so PCI would be the reference category. And so this is the analysis that we did. And so what I want to go through here is sort of the details of how you need to think about propensity scores using the inverse of the propensity scores in comparing the two groups. Okay, so in comparing the, or computing the propensity scores, this figure shows the distribution of propensity scores to be assigned to bypass surgery. So the distributions here for both groups, you can see that there's overlap there, actually a good amount of overlap between the distributions. And this is actually quite necessary. They have to have some overlap in order for the IPW method to actually work. So, so this is the first thing you sort of have to do, is to see, when you calculate the propensity scores, what's the distribution in the two groups. And if you get distributions that look like this, then you're in big trouble, because that means your propensity scores are not really identifying patients that are similar in most every way, or in a lot of ways. So this is why it's necessary to look at the propensity score distributions and compare those. Okay, so then this is our table 1, where we have the unadjusted data and then the data adjusted for IPW by IPW. So you can see, now this study was done several years ago. Had I sort of known what I know now, I would have used standardized differences here instead of p-values. But you can see that in the unadjusted data between the CABG and PCI, that there's virtually statistical significance for every variable except a couple here. And of course that, is would be expected, because with this large of an N, most of sample sizes, most every difference is going to be statistically significant. So after applying, though, the inverse of the propensity score, then we see that we have made the two groups, the two intervention groups, much more equal. And so when we did this, then in comparing these with either Ttest or chi-square, that none of them are statistically significant except for one down here, if you count that, which you can see that that's in terms why standardized differences here would be a much better, because 53.2 versus 53.8% isn't that big of a difference. But so anyway, you can see that what the intent here then is to equate these groups on all of the variables that we have available to calculate the propensity score. And so these are the results in terms of the survival in the two groups. So you can see here that there's an initial benefit for PCI, but the long-term benefit favors the bypass surgery. And so having adjusted for all the potential confounders, then we would have pretty good confidence that these results are accurate or meaningful. Now, the question, of course, then that a lot of studies don't answer is the idea of prevalence of a unmeasured confounder. And so in this study, we actually did a simulation to show what would happen if there was an unmeasured confounder. And so what we have here, so the lines here are different percentages of a confounder in the cabbage group from 5 to 40%. And so the way that you interpret this is, and they have an example here in the legend, if the confounder was present in 10% of the cabbage, which would be the green line here, then and if it was in 35% of the PCI group, and increased the hazard ratio, the risk of death, by about a factor of 2, which would be right down here somewhere. Then what that means then is that confounder alone could account for the observed differences in mortality. It's important to do this because you want to be able to show that what you've done, that the in terms of trying to account or to make the group similar, that the presence of an unmeasured confounder would be relatively small or relatively large, however it turns out. Okay, so in summary then, our propensity score is a probability of assignment to a group. The probability of assignment to the other group is just 1 minus that probability. And we get that probability from logistic regression equation. And the hope is that we get a pseudo-randomized sample. And then the probability, as I mentioned before, is just you solve the logistic regression equation and then take the exponent of the regression equation divided by the exponent of 1 plus the regression equation. So in matching, as we pointed out, we want to match A with B, patient in A with patient in B by the closest propensity score. But we need to specify how close or the precision we're going to match our matching criteria. So is it going to be 10 to the minus 3, 10 to the minus 6? However, regardless of what criteria or precision you choose here, it turns out that about a third of the sample can't be matched. And so that's one of the primary drawbacks, maybe the primary drawback to propensity score matching, is that you lose quite a few of your sample of your patients or subjects in your study. Inverse probability weighting, we calculate the propensity score, and then we use the inverse of that to weight our analysis. And the advantage here, of course, is that we can use all patients in the sample because everyone has a propensity score, and you then are able to use everybody. So these are some of the references. The first one is the study, the IPW study I went over. The second one is a really nice summary of discussing propensity scores, their use and limitations. And then the third one, I want to just mention a little bit about this because, entitled Why Propensity Score Should Not Be Used for Matching. And their study or their paper is quite interesting. It's a bit on the technical side, but if you're interested in it, I think you can get a lot out of it. And I think basically what it boils down to is that in matching, you can't you don't have all patients available for the analysis, and so you end up with what they argue is that if you try matching, that it may end up with a real imbalance between the two groups, because you have had to eliminate a fairly large number of patients. So I'll stop there and then open it for any questions that anyone might have. So I believe we use the question and answer, the Q&A for that, if you have any. Okay, if there are none, or if you think of some later on, you can email me. And I want to thank you for your participation again. And so we'll see you again in April. I'm going to do a session on cost-effective analysis in either randomized or in observational studies. Okay, so then thank you.